Hi, and welcome, scientists. We have 3 minutes to complete our do now. Please do so silently and await further instruction. Please do not touch the lab materials in your station. We will go over those after our warm-up. All right, so our do now warm-up question was, what is the powerhouse of the cell? So Jamie, go ahead and tell us what you put down. Mitochondria is correct. And everyone remember from when we first started learning about cells that the mitochondria is where energy is produced in the cell, which is why it's called the powerhouse. I noticed some other students putting down mitochondria as well for their answer. So good job, everyone, on our warm-up today. Carl, would you mind reading our learning target, please? Great, thank you. And just so we're clear, I'm going to read it again. I can observe the enzyme catalase break down hydrogen peroxide on organic materials. So we're going to talk today specifically about the enzyme catalase, and something for you all to keep in mind is that catalase is an enzyme that is found in most organic materials. And so you'll notice a couple of things in your lab today that are going to be our representatives for organic materials. You should have a tomato. You should have a potato. And then you should also have hydrogen peroxide as well as a dropper. And then I forgot one of our organic materials is chicken hearts. So we have some chicken hearts as well for organic material. And then acting as our control for our experiment, our inorganic material represented today by our coin. So each of you should have those in your lab area. You should also have a cutting utensil. Please do not touch the cutting utensil until directed to do so. And as always, we should have safety glasses or safety goggles, some kind of eye protection as well. So today we're going to be doing a lab observation using the organic materials in your station. You should have a coin in your station also, which represents the inorganic material. And today we're going to be working in pairs after I go through a short demonstration. So what I'm going to do first is put some hydrogen peroxide onto the unbroken skin of our potato and the unbroken skin of our tomato, as well as some hydrogen peroxide on our inorganic material. And we're going to save the chicken livers, chicken hearts, chicken gizzards, whatever you have. We're going to save that for step 3. So the first thing we're going to do is we're going to take some hydrogen peroxide and put it on our tomato. Unfortunately, I don't have a dropper, so I'm going to just be using a teaspoon, and I'm going to pour some directly onto the tomato. You guys notice it just kind of rolls right off. Same thing with the potato. It kind of just rolls right off. And then we'll put some on our coin. And it kind of just sits there. Doesn't really react. Doesn't really do anything. Okay? So that's step 1. We have put hydrogen peroxide onto the unbroken skin of our tomato and our potato. And so step 2 we're going to do together. So with me, you are going to pick up your cutting utensil and make a single cut in the tomato away from the hydrogen peroxide, and the same thing on the potato. So make sure you get a spot that doesn't have hydrogen peroxide on it and make a cut in the potato. Go ahead and put your cutting utensil back down. We'll be finished with that. And then we're going to bring over our chicken livers as well. Now, as you're in your partner groups, what I want you guys to do is add hydrogen peroxide to the cut that you made on the tomato, the cut that you made on the potato, and to the raw chicken innards. So I'm going to do a little bit of each. I'm going to put some hydrogen peroxide on the heart and on the gizzard. All right, and I'm going to wait. And I'm going to do the same thing on the tomato. And I'm going to do the same thing on the potato. All right. And so in your partner groups, you're going to write down your observations on your observation sheet that's also provided with your lab materials. And one thing that we'll notice right away is on the chicken livers, we see the hydrogen peroxide bubbling up. And so that's what we're really looking at. And now it's starting to happen on the potato, on the cut as well. You see the white hydrogen peroxide bubbling up. And so what's happening there is this enzyme catalase is reacting with the hydrogen peroxide and turning it into oxygen bubbles on the organic material. And then we'll notice also for a final observation that our inorganic material doesn't have any kind of reaction. Thank you, scientists, for coming to our lab today. And always remember that knowledge is power.